You take it away, Tom.
Good. Thanks, guys. Jeff, Thomas, it's great to be here. I'm Tom Vasicek, CEO of Gila Therapeutics, where we're developing completely safe and highly effective therapies for obesity and age-related weight gain. So, as Larry was saying, there's a huge problem with type 2 diabetes. The principal cause of that is the obesity epidemic, which preceded its sweep through the globe. And this is because we have evolved an incredibly powerful system, psychological and physiological, for acquiring and storing energy. 5,000 years ago, every human had to face times of food insecurity, and there's a very powerful system for enabling us to store energy when it is available. Today, we have more highly palatable, energy-rich food than we know what to do with. And in this environment, obesity is a huge problem. A few of us, a very small percentage, are actually able to control our weight with the large amounts of exercise and Herculean efforts to control the timing, the types of food, and the amounts that we eat. But for the majority of the human population, that's not possible. There's a huge number of people suffering from overweight and obesity worldwide, over 2 billion people. And for them, for most, dieting and exercise just don't work, either because they can't or they won't or whatever. And it's not a moral failing. Unlike aging, which we can still debate whether it's a disease or not, we really need to consider obesity as a disease, to destigmatize it, to enable people, to encourage them, in fact, to seek treatment. Currently, the treatments that are out there, as Stephen Smith said yesterday, just don't work that well. There's a huge unmet need currently for effective and safe treatments for obesity. All of the approved drugs out there don't work that well, and they all have serious side effects. They're not widely accepted by patients, and physicians are loath to prescribe them. The only really effective treatment, as Larry pointed out, is bariatric surgery, and it comes with a host of problems that he outlined. But as Stephen Smith pointed out and others have talked about, many of you are familiar with, the principal mechanism through which bariatric surgery works is not through malabsorption or calorie restriction, but by the alteration of our physiological state, the enhancement of the release of the very hormones that normally regulate our hunger, satiety, and energy metabolism. We found out more than 20 years ago, I was at Millennium Pharmaceuticals in the 90s, working to clone the genes for all these hormones, their receptors, and everything in between that we could possibly use as a therapy for metabolic diseases. We found a few years later that raising the systemic levels of these hormones could result in a significant reduction in food intake. Rachel Batterham here in 2003 published this New England Journal of Medicine article showing that a single injection of one of the hormones, PYY, could actually lead to a 24-hour reduction in food intake of 25%. We later found that no matter what we did in raising systemic levels of any one of these many hormones that regulate satiety, for instance, it also caused nausea. These hormones are made by the small intestine. Bariatric surgery sends nutrient-rich food to the small intestine more rapid, resulting in a more rapid and a more dramatic release of the hormones into the systemic circulation, where they go to the brainstem and the hypothalamus, and also signal via the vagus nerve to both these areas, generating satiety and nausea, both regions. I was at Medtronic at the time, delivering the hormones with insulin pumps, believing that we could really find a safe and easy way of modulating the hormone levels and not causing the nausea. But in fact, that wasn't the case. all those programs at the big pharmas and ours at Medtronic were stopped in about 2008. But about that time, at the University of Florida in the laboratory of Sergei Zolotukhin, Andres Acosta had found that when the blood levels rise after meals, blood levels of these hormones, they also appear in saliva, and that application topically to the tongue of these hormones could actually induce satiety and reduce food intake in animals with no systemic uptake. And long-term treatment showed significant weight loss. The trick was getting mice to enjoy taking these hormones. They used a mechanism using a small cosmetic sprayer, which worked pretty well. And it allowed them to do long-term experiments. You can see in one of the middle panels here. In the short term, here the two bars show spraying water and spraying PYY in this case on the tongue resulted in a significant acute food intake reduction. And over the course of 20 days, could prevent weight gain in a lean mouse model on a high-fat diet. Significant reduction in the amount of weight gained by these animals. To enable longer-term studies, they made adeno-associated viral vectors expressing the hormones and injected them into the salivary gland. And once those vectors began producing protein, we saw a significant reduction in weight here in an obese mouse model, diet-induced obese mice. Over the course, in this case of 8 weeks, lost 23% of their body weight. And here, over 25 weeks in a separate experiment, and again, lean mice on a high-fat diet, in the green fluorescent protein control mice, gained significant weight over this time, whereas the PYY expressing mice gained very little weight, significantly less. This could also be done with GLP-1, here showing acute food intake reduction with GLP-1 and receptor agonists exenatide. And here the combination, again, in AAV vectors, each hormone separately preventing weight gain, and the two together significantly reducing weight gain. So this led to a new paradigm, a completely new idea for drug delivery. No one had ever exploited topical lingual delivery for any therapeutic approach. This is not sublingual delivery. We're not looking for absorption. The receptors are present on the tongue and they wire directly to the satiety center in the brain via the 9th cranial nerve, the glossopharyngeal nerve. There's no activation of the nausea center. And we can in fact get full activation of these brainstem and hypothalamic regions for satiety, more than you can get with an injection and more than you'd ever get with a large meal, because we can actually saturate the response. There are enough receptors on the tongue and the bandwidth of that nerve is big enough that we can fully saturate the brain. And we've shown in imaging studies, behavioral studies, in numerous modes, and I'd be happy to show you those data, this effect. And we've in fact gone on to show this in our phase 1 clinical trial. So now we've got an exclusive license from the University of Florida for this intellectual property. And we've continued to expand that with formulation patents, combination drug patents, and a number of other modalities. And we've gotten our IND and completed phase 1 clinical trials. We've got a great team together. We've actually executed our phase 1 trial from first funding through all the preclinical work and CMC on $4 million in only 18 months. And now we're set to do phase 2. To review our phase 1 results, though, we fully met our primary endpoints. It was absolutely safe and there were no tolerability issues at all. No systemic absorption and no nausea. And oh, yes, Thomas? 4 minutes? Thanks. I'm going to wrap up. So we also saw a statistically significant increase in satiety using visual analog score questionnaires, asking questions like, how hungry are you? Or how full do you feel? We saw a 20-point reduction from placebo. That's twofold greater than separate studies done by Novo Nordisk with liraglutide, the leading drug on the market today for weight loss. So here is the actual output of this visual analog score questionnaire. Subjects arrived in the clinic starving. They had actually not eaten since the night before. We didn't give them much for breakfast. And so by lunchtime, they were really hungry. They're scoring really high. This is the overall appetite score, a mathematical combination of all the questions on the questionnaire. Everybody's very hungry. And so immediately after getting their first dose, they got a huge buffet meal, all they could eat. And for the next 30 minutes and the 60 minutes, they felt pretty full, whether they had gotten placebo or the drug. But after 90 minutes, and certainly after 120, subjects felt significantly more full, more satisfied on the drug than on placebo. This is just showing the Novo Nordisk data on the left, a 10-point change in this visual analog score questionnaire. And in our study, a separate study, a 20-point change with 3 different doses. We were actually able to saturate the system and go as much as we want another dose higher than this, 1,000-fold higher. So we reached the plateau, which ramped up here in the lower dose range. So now, as I said, we are ready to move on to our phase 2 clinical trials. For that, we need to do long-term toxicology and more CMC to develop a commercial formulation. And our studies involve a caloric intake study with our current liquid formulation. And later, when we have the oral dissolving tablet formulation that we're working on, phase 2A and phase 2B studies in healthy obese subjects without diabetes and in subjects with obesity and diabetes. These are some of the formulations in our intellectual property. The ODT is the one we've settled on. We've got about 24 others. And we have a number of hormones in our pipeline. Pipeline consists of several different of these hormones and combinations in a number of indications. Our team, as I said, is a great one. I'm really thrilled to be working with them all. And I look forward to meeting you all in the break room and following up with any questions that you might have. Thanks very much, Thomas, for the warnings. And Jeff, if you have any questions, fire away.
No, thank you very much. And just please remind us how much you're looking to raise and where that would get us after you raise it.
Raise $22.5 million, which will get us through those 2 phase 2 studies that I mentioned.
Great. Excellent. Well, thank you very much.